Dear students, let us learn about vitamin K, a fat-soluble vitamin, in this session. Vitamin K is one of the most essential fat-soluble micronutrient. It is required for a unique post-translational chemical modification of small group of proteins. It also exhibits calcium binding properties. One of the most significant functions is its unequivocal role in blood coagulation. It is derived from the Danish word coagulation. Vitamin K actually belongs to a family of compounds generally referred as quinones, that is a phyloquinone and menaquinones. Phyloquinone is widely distributed in plant sources, while menaquinones are found only in animal sources. Vitamin K can also be synthesized from intestinal bacteria. Menadione is a synthetic form of vitamin K. Liver serves as a major storage site for vitamin K. The liver stores are rapidly broken down and are eliminated from the body. Thus, the body stores are known to be depleted quickly. This module would help you to understand the origin of the nutrient, know the major food sources, learn about its physiological functions, metabolic pathway, and adverse effects of deficiency. First, let us learn about the structure and nomenclature of vitamin K. This possesses a common 2-methyl-1,4-naphthoquinone structure referred as menadione. It also contains a side chain at the third position. This compound is not a natural constituent of foods. It has the ability to add on geranyl side chain at the third position and yields menaquinone 4 or MK4. In this regard, menadione can be considered as a pro-vitamin K. Naturally occurring forms of vitamin K possess the same naphthoquinone ring similar to that of menadione, but they would tend to differ in the structure of side chain at position 3. This could be traditionally classified into two categories in accordance with the origin of synthesis either by plants or bacteria. In plant origin, vitamin K, the major form is termed as phyloquinone or vitamin K1, which comprises the same phytyl side chain as that of chlorophyll. Bacteria also synthesize a group of compounds called menaquinones and is termed vitamin K2. The food sources of vitamin K is the phyloquinone is widely distributed in nature. Green leafy vegetables are known to be the richest sources and they contribute significantly to the dietary intakes. The relative concentration of phyloquinone in vegetables reflects their direct relationship with photosynthetic tissues, and highest values are found in dark green leafy vegetables, namely the kale, parsley, spinach and cabbage, with intermediate amount being found in broccoli, Brussels sprouts and lettuce. Significant amounts are also found in non-leafy vegetables like green beans, cauliflower and cucumber. Vegetable oils, namely the soybeans, rapeseed and olive oils, also known to contain relatively higher amounts in the range of 50 to 200 microgram per 100 gram. Fish and skeletal muscles, cereals and beverages are found to contain only negligible amounts. Cheese is a fermented milk product and this is known to contain two major types of menaquinones, that is MK8 and MK9. Their concentration ranges from 5 to 10 and 10 to 20 microgram per 100 gram respectively. These might be thought to have been derived from the bacterial starter cultures adopted in the process of fermentation. Because molds are not capable of synthesizing menaquinones and its content was estimated to be less than or equal to 1 microgram per 100 gram. The liver of ruminant species have been estimated to contain 10 to 20 microgram of menaquinones per 100 gram. The vitamin K2 is usually considered as a typical of the menaquinone series because it is not generally synthesized from bacteria. It is abundantly found in animal tissues and eggs. Apart from these, bacteria that are normally known to populate the microbial ecosystem of the human intestine are proposed to synthesize menaquinones. After knowing the sources of vitamin K, next look into the physiological functions of vitamin K. This has an essential role in the synthesis of substances needed for blood clotting. Prothrombin factor 2, proconvertin factor 3, plasma thromboplastin component factor 9 and Stewart factor and factor 10. Vitamin K is needed for the conversion of prothrombin to prothrombinogen in the liver. This is mediated by the conversion of glutamic acid of the protein into a new amino acid called gamma carboxyglutamate acid. This in turn catalyzes the conversion of prothrombin in the blood to fibrinogen, a factor involved in the formation of fibrin in blood coagulation. The level of prothrombin in the blood is a major determining factor in the rate at which the blood will clot. The blood will clot only if the soluble protein fibrinogen is converted into fibrin. Thrombin is known to be involved in catalyzing the conversion of fibrinogen from fibrin. There are mainly two pathways, extrinsic and intrinsic, which play a role in the generation of prothrombin and thrombin for blood clotting. Let us look into the intrinsic pathway. The process of coagulation begins with the absorption of factor 12 into collagen. The factor 12 gets activated into 12A. This precedes the process of cleaving the factor 11 to yield an active compound 10IA. This in turn cleaves the factor 9, which is highly dependent on vitamin K. The carboxylation process binds with calcium and phospholipids that are formed from the aggregation of platelets. This helps for the conversion of 10 to 10A, which is also a vitamin K dependent factor. 10A in turn leads to the hydrolysis of prothrombin or the factor 2 to thrombin, the factor 2A. This completes the conversion of fibrinogen to fibrin, resulting in the formation of clot. Next, we will see about the extrinsic pathway. In case of tissue injury, the tissue thromboplastin activates the factor 7. The factor 7A is vitamin K dependent. Similar cascade of reactions are known to occur, which was described in the intrinsic pathway. This ultimately results in the synthesis of thrombin from prothrombin. We will look into the role of vitamin K in bone health. Physiological and observational evidences have suggested that vitamin K plays an important role in bone growth as well as the maintenance of bone density. Supplementation of vitamin K has shown to promote osteotrophic process via slowing down osteoclastic processing through calcium bonding. A combination of in vivo and cell culture experiments have found that MK4 helps to reduce bone losses caused by either withdrawal of estrogen or corticosteroid treatment. The formation of osteoclast-like cells in bone marrow derived cultures is known to be inhibited by MK4. Next, in cancer, studies have determined that MK4 has the greatest potential of inhibiting the growth of certain types of cancer cells from those of liver and gut and some types of leukemia cells, either by inducing the cell differentiation or by apoptosis. In vitro studies have demonstrated that MK4 menadione and menadione related analog, that is vitamin K5, was known to reduce cell proliferation and anti-tumor effects in colorectal cancer cell lines. MK4 has been known to suppress the development of hepatocellular carcinoma among cirrhotic patients. It also reduces the incidence of recurrence of hepatocellular carcinoma after resection. Next, we will see the role of vitamin K in inflammation. There exists an inverse relationship between vitamin K and inflammatory response. Studies performed on animals have showed that vitamin K deficient diet resulted in an increased expression of genes that are involved in acute inflammatory response, while supplementing vitamin K was shown to suppress the inflammatory response. Next, with regard to the oxidative damage, it has been evaluated to have a definitive role in preventing the oxidative damage to oligodendrocyte precursor cells and fetal cortical neurons. Oxidative damage is known to be the major factor leading to cell death in brain disorders such as perinatal hypoxia or ischemia. Under these conditions, the pre-myelinating oligodendrocytes are known to be damaged by reactive oxygen species. Vitamin K has also been reported to possess free radical scavenging activity and thus would help to suppress lipid peroxidation in liver. Next, we will see its role in sphingolipid synthesis. Sphingolipids are nothing but a group of lipids found in large amounts in brain. The importance of vitamin K for their synthesis was evaluated by a study conducted on bacteria. When vitamin K requiring bacteria were grown in vitamin K depleted medium, it exhibited defects in cell envelope. This was mainly caused due to a reduction in the activity of 3-keto-dihydrosphingosine synthase. It is known to be the first enzyme in the sphingolipid pathway. Galactocerebroside sulfotransferase, a brain enzyme said to be sensitive to vitamin K depletion. MK4 supplementation was shown to be beneficial for restoring the activity. MK4 is the major principal form of vitamin K that is present in the brain of humans and animals. It is known to be present in highest concentrations, particularly in regions having higher amounts of sphingolipids. Thus, it could be said that MK4 is a highly physiologically active form and has been implicated in the synthesis of structural lipids in vivo. The mechanism of action of vitamin K in mediating these effects is still unknown, but it has been postulated that it is one of the vital nutrients in the brain which is required for the synthesis of lipids needed by myelinated cells. Thus, under conditions of vitamin K depletion, it is manifested as behavioral disorders attributed to vitamin K deficiency, or it could be an effect induced by warfarin treatment. We will see how vitamin K is absorbed. Dietary vitamin K as phylloquinone is absorbed in a chemically unchanged form from the proximal intestine. It gets solubilized into mixed cells comprising bile salts and the products derived from the pancreatic lipolysis. The extent of absorption of phylloquinone among healthy adults has been reported to be around 80%, and the diet containing green leafy vegetables like spinach would tend to decrease the rate of absorption to less than 10%. It exerts a strong association with the thylakoid membranes of chloroplasts. In the intestinal mucosa, the vitamin is incorporated into chylomicrons, which secretes into the lymph, and via the lacteals it enters into the bloodstream. The circulating phylloquinone is rapidly cleared at a constant rate along with continued association with chylomicrons and the chylomicron remnants. More than half of the circulating level of phylloquinone is reported to be still associated with triglyceride-rich lipoproteins even after an overnight fasting. The remainder would get equally distributed between LDL and HDL. The major circulating form of vitamin K is the phylloquinone. In plasma, MK7 is found to be present at a lower concentration, and its lipoprotein distribution has been observed to be similar to that of phylloquinone. Dietary vitamin K in the form of chylomicron remnants has been thought to reach liver and various tissues including bone marrow. Liver is the principal site for the synthesis of vitamin K dependent coagulation proteins and is known to have limited capacity for storing phyloquinone. Hence, the hepatic reserves of phyloquinone are estimated to be 1000-fold lower than those of vitamin A reserves, while the estimated dietary intake is just about 10-fold lower. The tissue-specific vitamin K distribution patterns have been identified in humans and rats. Vitamin K accumulation in heart, brain and pancreas is suggestive of the fact that it might have an unrecognized physiological function associated with these organs. Phyloquinone is mainly absorbed by an energy-dependent process in the small intestine, whereas menaquinones and menadione would be absorbed in the small intestine and colon by passive diffusion. Its absorption is mainly dependent on several factors, including the presence of minimum amount of dietary fat, bile salts and pancreatic juices. The metabolism of vitamin K yields mixtures of vitamin K vitamers, and thus the tissue are found to contain mixtures of vitamin K despite consuming a single form. Majority of the tissues are estimated to contain phyloquinones and menaquinones only. Let us look into the metabolism of vitamin K. This occurs in a series of processes of bacterial dealkylation and realkylation converts phyloquinones to menaquinones prior to absorption. The metabolites that are produced by side chain shortening and oxidation are excreted in feces via the bile as glucuronic acid conjugates. This results in catabolization of phyloquinones and menaquinones. Menadione would be metabolized at a much faster rate and is primarily excreted in the urine as phosphates, sulfates or as glucuronide derivatives. We will look into the stores and distribution of vitamin K. Human liver comprises about 90% menaquinone and 10% phyloquinones are stored in human liver. Evidences indicated that liver stores of phyloquinone are very labile. Under severe dietary depletion, liver concentrations would tend to decrease to about 25% of its initial value within a span of 3 days. The hepatic stores of phyloquinone among different population groups is very scarce. We will see how vitamin K is excreted. Extensive metabolism is known to take place in liver and it is mainly excreted in urine and bile. Carboxylic acid with 5 and 7 carbon side chains have been identified as the major human excretion products. These are excreted in urine as glucuronide conjugates. The biliary metabolites are initially excreted as water-soluble conjugates. These in turn become lipid soluble while passing through the gut via deconjugation of the gut microbial flora. Next we will see what are the effects of deficiency of vitamin K. Deficiency in adults resulting due to hepatointestinal disorders impairs the absorption and utilization of the vitamin. The excessive use of oral anticoagulant drugs such as warfarin, which is used in the treatment and management of thromboembolic diseases, is considered as a major causative factor for the occurrence of deficiency. These anticoagulants would tend to reduce the circulating levels of vitamin K dependent clotting factors to some predetermined range. This can be evaluated depending on the clinical condition of an individual. The next one, the hemorrhagic disease of the newborn. This was discovered in 1894 by Townsend. It is described as a self-limiting bleeding disorder which is seen among newborns and it is considered to be of unknown origin. After the discovery of vitamin K, it was realized that HDN had similar features of a syndrome among newborns in the first week of life and it was shown to be caused by a deficiency of vitamin K. Up to 1960s, HDN was considered to be a problem of first week of life. There are mainly two forms of deficiency that have now been recognized: late HDN and early HDN. Late HDN has been said to be a potentially more serious syndrome which is known to reach its peak between 3rd and 6th week of life. Early HDN is known to occur during the first 24 hours, and this is mainly caused by antagonist drugs taken during pregnancy. Late HDN is known to occur very rarely, and its prevalence rate is about 4 to 70 cases per 1 lakh live births, and is known to be a significant cause in infant morbidity and mortality worldwide. Estimated 50% of the cases of late HDN is known to cause intracranial hemorrhage leading to death or severe and permanent brain damage. The major risk factors for the occurrence of both these syndromes are exclusive breastfeeding. Since human milk is known to contain relatively low concentrations of vitamin K, and intake of maternal milk in the first week of life has been reported to be the major risk factors. Poor placental stores have also been considered to be a probable risk factor. These reduced stores have been estimated to contain only phylloquinone, and the concentration of bacterial menaquinones are absolutely nil at birth, and it gradually builds up. Liver functioning abnormalities are found to be very common among infants who develop late HDN. These manifestations are often considered as mild and as self-correcting. No screening tests are available, and bleeding can occur spontaneously. Hence, the best option is to protect all infants by providing vitamin K supplements during the immediate perinatal period. A convincing body of evidences is available which implies that without vitamin K prophylaxis, infants are known to be at a greater risk of dying, or the brain may be damaged permanently due to severe depletion of vitamin K during the first 6 months of life. The use of multiple oral supplements has become very common instead of the single intramuscular injection generally given as 1 milligram of phylloquinone at birth. In order to minimize the risk, the American Academy of Pediatrics has recommended the administration of 0.5 to 1 milligram vitamin K to all newborn babies immediately after birth. There is also certain symptoms of toxicity of vitamin K. Oral intake of vitamin K has been reported to be free of toxic side effects. This has been demonstrated through clinical administration of phylloquinone at the rate of 10 to 20 milligram or greater levels. Patients suffering from chronic fat malabsorption can ingest doses of 10 to 20 milligram without any reported harmful effect. From the nutritional point of view, it is safe to avoid synthetic preparation of menadione or its salt among newborns. Vitamin K is known to lack intrinsic biologic activity. The unsubstituted third position is known to be highly reactive and has a strong association with the occurrence of neonatal hemolysis and liver damage. After learning about the toxicity, now we shall look into the recommended dietary allowances of vitamin K. And there is not much data available, and there is a very limited data on the vitamin K intake as well as its requirement. It exists widely in plant-based foods, and dietary deficiency is not normally encountered. The FAO WHO has suggested an RDA of 55 microgram for an adult. Now, to summarize what we have learned so far, we can understand that vitamin K plays a significant role in the maintenance of blood coagulation. It is commonly referred to as the quinones. It is found widely distributed in plant-based foods. Apart from facilitating blood coagulation, it also helps to maintain normal bone health, prevents cancer, inflammation and oxidative damage. Severe deficiency is known to be manifested as hemorrhagic disease commonly seen in infants. It is often regarded as a nutrient free of toxic side effects. Hence, adequate amounts should be taken through daily diet in view of preventing the problems associated with blood coagulation.